Okay. It's my pleasure to be here, and thank you very much for asking me. Up front, I want to say that I don't consider myself an expert in partnering with industry. In part, that's because I rarely have had direct interactions with the people with industry myself, although, as you'll see, the work that I do requires this interaction to take place, and I'm just going to go through it from my perspective. And please feel free to ask questions, and I hope this is worthwhile for you. I think the title is Lessons from a Slow Learner, which may be in part my personality, but it's also that I have a feeling that at each step along the way of what I'm going to discuss, I learn things painfully by making mistakes and then trying to learn from them. So with that in mind, I'm going to go forward. So I thought for the first few slides, I would just sort of set out an agenda when somebody like me thinks that they may have interactions that are ultimately going to require interactions with industry. So if you're in this situation, you're probably interested in developing a clinical trial for a particular disease. And as you think about this, there's a question of, do you know what medications or types of medications you're interested in? And are any of these medications going to already be approved from the FDA for related disorders? And or are you going to have to develop medications from scratch and go through an approval process? And do you actually know about the pathogenesis of the disease in question? And then this is a bit of a rhetorical question because it's hard to have you just reply directly to me, but you need to think about what industry wants to know before they agree to enter into a partnership. And in my case, the question is, are they interested in rare diseases? And I think the answer, as just briefly to mention a company like Genzyme, is yes, they are. And those interactions can be very fruitful. Now, the next slide just emphasizes a lesson that I've learned going forward with the CMT world, in that industry is willing to partner for the inherited neuropathies, but they are really interested in de-risking their investment. So if you, or if I had gone to a particular company to say I wanted to find a drug to treat Charcot-Marie Tooth disease type 1A, they would want to have known, well, what assays do you expect to use? And what are the disease mechanisms involved? So what cellular pathways are we going to have to look at? And what's the natural history of the disease that you want us to treat? How are we going to know if a treatment's been effective? And what experience do you and your colleagues have in doing trials or in working together? And you need to be able to answer these questions if you want a serious partnership with industry, at least in my experience. So again, I feel a little bit over my head portraying myself as an expert in dealing with industry to develop trials. So I thought the most informative way to do this webinar is for me just to walk you through what's happened to me in my approaches to this issue, and just use that as an example. So my main interest, as Sam said, is in the inherited peripheral neuropathies, which we call Charcot-Marie Tooth disease. And I'm going to spend just a few slides going over what I mean by those. So the inherited neuropathies are disorders of peripheral nerve that are caused by mutations in genes. And currently, there are over 80 genes that cause peripheral neuropathies on multiple chromosomes. So Obviously, I can't go to a company and say, "Oh, I want you to work with us to uh, uh, develop and test treatments for 80 different genes." And so let's take a look at what these genes actually do. So they cause peripheral nerve damage in two ways. First, they typically involve the myelin, which is in-sheath peripheral nerve, and those we tend to call CMT type 1, or they involve the axon, which we tend to call CMT type 2. But the most common form of CMT is where we spent most of our efforts, and this is caused by a duplication of a protein called peripheral myelin protein 22, or PMP22, and it's an integral myelin protein. And what happens is that the gene for this protein is part of a duplication on chromosome 17, so that people with CMT1A get an extra copy of PMP22. And this is a slowly progressive disorder, which gets into issues we have to deal with when we're trying to partner to develop trials. Because most patients are normal at birth, are clumsy as children, and slowly develop neuropathy throughout their life. And a minority of patients ever end up in wheelchairs. And these are just some of the initial symptoms, which I'm not going to read, but you can see. And these patients have uniformly slowed nerve conduction velocities. So nerve conduction velocity should be about 50 meters per second. In this case, they're 20 to 25. And uh, because they're in demyelinating peripheral neuropathies, there's secondary axonal degeneration. Amplitudes on the nerve conductions eventually begin to shrink down, and as a marker of axonal degeneration. And we're going to come back to that with our outcome measures, because that correlates better with disability than the speed of the conductions. And again, this is a dosage disorder, and so this is just a couple of slides taken from skin biopsies showing that if you look at myelinated nerve fibers, which I don't know if you can see my arrow here, but these are the myelinated nerve fibers, and you can see them coming up in the skin. And you can do immuno-EM, and you can look at these dots, which are myelin proteins, and you can quantify the number of dots so that in people with CMT1A, they have too many dots. So they have too much PMP22. And the treatment strategy has been to try to identify medications that reduce the amount of PMP22. And that's going to be the sort of the overriding theme of what we're going to be talking about as we try to take this to clinical trials. So that's a sort of a brief introduction. And what I'm showing you here is a slide that's relevant, because I have to go to a meeting with other investigators this coming week with Sanofi Genzyme to look at uh, candidate medications that would decrease PMP22 levels and could lead to treatments for people with CMT1A. And there's been interactions with CMTA with other companies, but this is a particularly nice one to me because of all the work done by many people. In addition to me, they've already screened 2 million compounds, which we're trying to narrow down now to identify compounds to uh, uh, hopefully treat CMT1A. And this is the agenda for this discussion, and these are some of the issues that we're going to talk about in the rest of the webinar. So they want me to talk about a clinical trials path. They're using high throughput screen assays that they didn't develop, as we'll come to, but they're going to be able to use now. They're going to want in vitro myelination assays. They're going to want information about stem cells to use in cellular assays. They're going to want in vivo models, and they're going to want biomarkers. And without these, the project doesn't go forward. And a lot of these had to be developed in advance of working with Sanofi Genzyme. So again, they're willing to make the effort and allow their compounds to be screened, but only in the situation that the people like ourselves going to them have de-risked the agenda. So let's go through those items one at a time. And the first one I'm going to go through with is trying to use and develop clinical outcome measures, because I think that that has the most relevance for this audience. And again, I'm not going to tell you the correct way to do it. I'm going to tell you what struggles that we've had to go through to actually get one of these that works, and works in a way that we can detect change in a reasonable period of time to do a clinical trial. So the primary outcome that we're going to use in this trial is likely to be the CMT Neuropathy Score version 2 Rash. And I'm going to tell you how we got to that. So I feel like I'm repeating myself here, but this is just, again, the background of CMT 1A. And a reminder that the impairment correlates with axonal degeneration, not with the speed of the nerve conduction velocities, and amplitudes from the nerve conduction velocities can be a surrogate marker. So this work started basically a decade ago with a collaboration with Mary Riley from London, Davide Parison from Italy, and myself as we began to develop what we called the CMT Neuropathy Score. And we didn't develop this from scratch. We looked around. None of us had really expertise in outcome measurement development, but we wanted to find a tool that was easy to do, not time consuming, and that could be done in a reproducible way. And we didn't want to reinvent the wheel. So we looked and we chose a tool that was developed by David Kornblath and Vinay Chaudhary at Johns Hopkins for chemotherapy neuropathies, which is called the Total Neuropathy Score. And it was a way to measure length-dependent, impairment in length-dependent neuropathies, in their cases where there was a lot of sensory components. And it was a composite score, and we tweaked it to make the CMT neuropathy score. And for some reason, it didn't show up there, so I'm jumping a bit. Just bear with me for a second. I want to see if I can find the score. I'm sorry for this. Well, this is a updated version, which I'm going to come back to, but at least I can walk you through this. So this is the version 2 of the CMT neuropathy score, and you see it has 9 components. The first 3 are symptoms, like the sensory symptoms, the motor symptoms in the legs, motor symptoms in the arm. The next 4 components are things that we measure on the exam, and the bottom 2 components are the size of standard motor and sensory amplitudes that we look at in the nerve conduction velocities. And the higher the score, the more problems somebody has. The maximum score is 36. And so we developed this score and tested it. And I'm going to move back a couple of slides now. I apologize. And we published this in Neurology in 2005, and then we tested it in about 80 patients over 2 years, and we found that we could detect a progression of 1.3 points over 2 years that was significant, and we published this again. And we were feeling very proud of ourselves, and we then went and undertook clinical trials in CMT1A, both in Italy and the United Kingdom, and also in North America, with the CMT neuropathy score as our primary outcome measure. And what we found was, first of all, the ascorbic acid, which was the agent we used to treat, didn't have an effect. But what we also found is, if you look at the bottom tracings here, that not only was there no change in patients who took ascorbic acid, but there was also no change in the placebo group over the 2-year period. So what we had seen in our natural history studies, which we tried to do the best that we could, was not the same as what we actually found when we did the trial. So our lessons here were that the placebo did not equal the natural history. So we got different results with those two. And we also wondered if the CMT neuropathy score was sensitive enough to change in a slowly progressive disorder like CMT1A. And we wondered whether there was too many floor and ceiling effects, and we wondered whether there was a spread across the full range of scores. So again, we had started in 2005, and we are now up 4 years later to trying to figure out ways to improve this outcome measure. And we held a meeting in Naarden, ENMC meeting, where we brought people who were experts from all over the world together to try to improve this score. And what we did was make the version I showed you before, where we tweaked the way we did sensory testing. We put a quantitative vibration testing in using a Rydell tuning fork. We tried to figure out ways to make the score more sensitive. And I can tell you, now several years later, and looking at a very large number of patients, 300 or 400 patients, that this score still does not detect much change within a 1 to 2 year period, which you need to do for clinical trials. Oops, and I think, let's see what I did here. And well, these were some of the issues that we addressed at the Naarden conference. But what we also learned going forward was something that we had not known about before, which was rash analysis. And I'm going to try to explain how somebody like me interprets rash analysis, and I still don't consider myself an expert. But when we looked at our primary outcome measure, the CMT neuropathy score, we saw that patients tended to clump in the middle here in terms of their severity. And that our CMT neuropathy score was pretty good at picking up people with very mild CMT, and it was pretty good at saying who was very severe. But if you weren't in one of those two groups, you tended to clump in the middle, so that any change between the middle to severe or from mild to the middle would be lost unless it was a marked change. By submitting our score to Rash Analysis, we were able to linearize it, and we're excited that I think in Quebec at the CMT and PNS meetings in a couple of months, by using the Rash Analysis, we're now able to detect change within a year to 2 years, which will actually allow us to go forward with this as an outcome measure. But it took 5 years or more to develop this, in part because of a lack of knowledge by the people who were working in the area. And so I think your course in really training people how to develop clinical trial outcome measures, and have this be a scientific approach is like really essential to move this process along in a faster way than I did it. Another thing that we learned going along was that a lot of the changes that occur in patients with CMT1A and other forms of CMT occur within the first 2 decades of life, and that our CMT neuropathy scores, first of all, required cooperation that younger children weren't able to easily do for us. And then also we had to deal with the fact that children grow and develop, so that what's normal for a 15-year-old may not be normal for a 5-year-old. So we were fortunate to have people like Rick Finkel, who's now at Nemours, and Josh Burns from Sydney, who were able to help us design a outcome measure in children that we're now using and pleased with. And it's just a picture of Rick on the left and Josh on the right. and we work to make this score functional to children's daily activities. So this is just to show you the score, and I'll go through it in a little bit of detail, but mainly I think the issue is that it's adaptable to change and normalized to age. So this score requires us to know the age, gender, height, and weight of a child so that we can normalize it to sex and age. And this is a much more functional score than a composite score, as I talked about before. And it has tools of hand dexterity, quantifiable strength measurements. You can see the sensation testing with the neurotips and a quantified tuning fork. That's a Rydell tuning fork. And the Bruninsk-Osiretsky equipment for looking at balance. The 6-minute walk and the long jump. And I think a key, and this was developed by Josh Burns, is this online tool where you put in the values from each child, and then by clicking on the icon down here in the lower right corner, it normalizes the score based on standard normal values for each of the components, so that you get a normalized score for age and sex. So this allows us now to be able to have a functional score which can be used in children between 4 and 21, which we're also finding detects change within a 1 to 2 year period. And this slide I just put in to remind me to say that for all of these scores, both the PED score and the adult score, that it's essential for us to cross-train with each other, and so that we get the same score on the same patients, and also that we have online teaching tools to ensure that everybody is instructed in exactly the same way and performs the exam in exactly the same way, or else like small progressive changes will be lost because of variability. Now, again, I'm talking about lessons that I've learned, and in 2005, even in 2008, I didn't even know there was such a thing as a patient-reported outcome measure. But fortunately, we've had people like Sindhu Ramchandran from the University of Michigan and other people who have actually been formally trained in developing patient-reported outcome measures. And this has been extremely important for us as we go forward. So Sindhu has developed and published the Disability Severity Index, and colleagues from the Netherlands have developed something called the Hereditary Motor Sensory Neuropathy Rash Outcome Disability Score. And this has allowed us to actually have the patient-reported outcomes. And this, as you probably know better than me, these are required, I believe, by the FDA for many clinical trials going forward. And to say that you're going to develop a patient-reported outcome measure sounds nice, but I just want to show you what it actually has entailed for Sindhu and Fleur, and also David Herman and others who've been working with this. So we're fortunate enough to have a consortium on inherited peripheral neuropathies, and one of the things that we have in this is a patient contact registry where there's over 2,000 people worldwide who are linked in with this contact registry. So that when Sindhu and people that I've been talking about put together questionnaires to try to determine what people who have CMT think is important for them, we have access to this to actually help define the instruments. And again, Sindhu and Fleurkamp have queried the patient contact registry to be able to rapidly develop these instruments. And these have both been published or in the process of being presented now. But again, I think the lesson here is to make sure that you have access to the patient population affected by the disorders that you want to develop the patient-reported outcome measures for, and that you have expertise in really designing these. And I think the promise set up through the NIH is really excellent here. And this is just slides. I'm sorry, I'm going through these quickly, but just showing basically about the contact registry. But I think the points have been made. And again, these are all various patient-reported outcomes, and I'm sort of sliding through these. These are just ones that have been done through our consortium. And again, I think I've already made the main point, so I don't know if it's worth spending more time on this. Another important tool that we've learned about is the necessity of quality of life instruments. And we have begun with studies using the standard SF36 instruments, which are generic across disease, but we've learned the need to develop disease-specific quality of life instruments. And so these are just one for adults by Nick Johnson on the top and Sindhu Ramchandra in the middle on CMT-specific quality of life instruments for children's. And again, to have these available for going forward to clinical trials has allowed us to actually be looked on very favorably by industry such as Sanofi Genzyme and others. Going forward for clinical trials, we've needed to have primary outcome measures, but we've also needed to develop other markers to look at disease progression. And for a slowly progressive disorder like CMT1A, this has been a challenge. These are some of the ones that we've been looking at. On the bottom, looking at changes in mRNA levels from skin biopsies has not shown much change over a 1 to 2 year period. We're looking at quantitative sensory testing, as you can see there. But the one that we're the most excited about comes from the MRC group in England, who have really done pioneering work with studies that look like we're going to be able to detect changes in fat fractions with MRI imaging within a year's period of time. This has been done entirely by the MRC group in London, and we're very excited about it. But again, this has been years in developing, but is extremely helpful for us when we go to industry to be able to have tools like this to talk to them about. This is just showing the negative results with the skin biopsy PMP, 22 mRNA, so I'm going to skip by it. So that's sort of where we are when we talk to companies about outcome measures. And I think that we can make a case that we have experience, we have a large number of patients, and that we have worked carefully to define useful outcome measures. My point is that it took us about a decade to do this, and all of the information that we have is really necessary for industry to feel de-risked. So hopefully it won't take people in this course a decade to get this type of information, but these are the kind of things that you need if you want industry to treat you seriously going forward in clinical trials. Now I'm going to just spend the rest of the talk just going through where we are in some of the other components of that initial schedule I showed you with Sanofi Genzyme, because all of that was done before going to the company. And it's because of having all of those in place that the companies look favorably on our group, I think. So for us to get back to the biology, we were looking for compounds that could be screened to identify reductions in PMP22. And to really do this in a high throughput manner required cell design. And so the first part of what was done is work that was done largely by John Severin at the Weizmann Institute and previously by Uli Suter in Zurich. And I don't want to go through too much of the molecular biology here, but for PMP22, between exon 3 and exon 4, there's a regulatory domain which Schwann cells use to drive expression of PMP22. And there's binding sites there for two transcription factors, SOX10 and EGR2. And what, in this case, John Severin did was he took this region and linked it to two reporter constructs that express various types of luciferase and used this, put it into a Schwann cell line and used this as an initial screen for compounds that could regulate PMP22 expression. Because if they decreased PMP22 expression, they would reduce the level of luciferase, which could be then detected. And this is just the same thing on a better slide. And again, these are the reporters, the firefly luciferase, and actually the beta-lactamase has been replaced by another form of luciferase. And this is another slide that's not showing, but what this is that this is the robotic system that's at the NCATS facility in Rockville, Maryland, where there's basically robots able to look at thousands of compounds at serial dilutions in cell lines made by John and his colleagues. And this enables screening of up to hundreds of thousands of compounds in a very short period of time. And it's because this system was in place and because the cell lines that John had put in place already were being used in this and had been tested, that a company like Sanofi Genzyme was interested in partnering on this project. And again, this is the critical path that was put in place before going to companies to screen candidate treatments for CMT1A. So to have the reporters, then to be able to take hits from the reporter screens and look by techniques like quantitative RT-PCR to look at PMP22 levels themselves, then to be able to take these to actually human lines aren't on here, but they're a part of it, and ultimately into animal models to be able to take to testing, to having all of that pathway set up in advance. When that's there, the clinical information is there, it's really then that companies really get excited. This is just a repeat. And this is just to comment, first of all, on where the animal models came from, because these are both the two, I think, best models, rodent models for CMT1A. But the reason I wanted to mention this also is that to assay treatments in rodents, and I think this also is related to a recent publication by Story Landis a couple of years ago, that you have to make sure that the power design is adequate for these and that it's done in a non-biased way. So what's happened with this is that the CMTA has made a partnership with a company in New Jersey or New York called Psychogenics, and all the animal models are housed there. Nobody from the CMTA or any of the investigators on the projects have anything to do with the valuation of the animals. And this facility is designed to do clinical trials on mice and rats. There is a excellent scientist named Larry Rabbits who has given presentations on sort of animal hospitals for this kind of study. And I think this really fits that bill. And so now when we go to a company like Sanofi Genzyme, not only are the assays have been developed, but the animal analysis is done in a neutral way, and everything's powered adequately so that there's confidence that this has been addressed in a way to really get true results. Now, this is actually a slide that I stole from Mario, who's one of the people on the call. And this is, I didn't have one for CMT1A, so I just stole this because this is for another form of CMT with a different gene. But the reason I'm putting this up here is that in negotiations with Sanofi Genzyme and other companies, they really like to have a human assay involved with human cells before they're comfortable going to trials in humans. And so developing iPSC Schwann cells from humans is an important part of what's been done, again, to keep, to have this pipeline going from an idea of lowering PMP22 levels to actually rational treatments for patients can be in place. So I just want to go back again to our initial agenda for the Sanofi Genzyme meeting. And these are the things that the company wants to discuss in at this upcoming meeting next week. And much of what's done here was not done by Sanofi Genzyme. It was done by investigators who wanted to develop treatments for CMT1A and getting things ready to go to a company. So Sanofi Genzyme basically, in a collaboration, took the assay from NCATS and John Severin and used it for their compounds in their screening system. And as I said, they've already screened 2 million compounds, and there's a number of hits that are going to be going forward with them. All the other stuff here is in place, and so they're de-risked. And I think de-risking companies, again, is the big issue in my experience with getting companies interested in going to clinical trials. So if you're involved with the kind of work that I'm talking about, this is the path that we've had to take. And I think with courses like this excellent course that you guys are in, being able to figure out ways to shrink what's taken me 10 years to do to a more reasonable time, I think is I think is like a really a goal of this whole course. So I think that might be my last slide. So I'm going to stop there, and I'm going to try to answer some of the questions. But thank you. So I don't know what happens next, if I just stay here or what happens.
Yes. Yes, thank you, Dr. Shai, for that presentation. There are some questions.
Would you like me to? Should I just go through those?
Yes, please.
Okay. So the first one is from Heidi, and why are patient-reported outcomes important? And I'll just answer that from my perspective, that the outcome measures that I talked about, like the CMT neuropathy score or the pediatric score, are measures designed by people like me who are scientists or physicians, but we're not the ones with the disease. And a question, which is reasonable, is if we detect and test a medication and it changes where a person feels a pinprick touch them or improves their ability to feel a tuning fork vibrate or changes the size of a wave on a nerve conduction test, has that really impacted their life? Because after all, what we want to do is to develop treatments that impact people's lives who have these diseases. So by having the patient-reported outcome measure, then you're starting to address issues that are relevant to patients. And ideally, you would have a treatment that would improve their life and quality of life. So I think it's in that spirit that the patient-reported outcome measures are important. Now, these patient-reported outcome instruments are complex and probably beyond my ability to talk about in just a few minutes here, but I think there's a need to make sure that there is changes in the biology of the disease as well. So I think that the patient-reported outcome measures are complementary to the, if I can use the term, biologically based outcome measures or scientist-based outcome measures, because there's advantages to both. But I think that for certain, the patient-reported outcome measures are necessary and more important than what I think the FDA believes that. The next question on here is from Will, and it's involving something that I'm involved with but didn't talk about here, which is when developing a biobank, what are the major considerations early on that should be addressed to strengthen its utility? And I think that this is an important question, and because biobanking takes a lot of time and effort and coordination. And if you just are putting a bunch of cells in a freezer and they're going to stay there for the next decade and nobody's going to know what to do with them, then you're wasting people's time. So for us, the main situation was developing fibroblast lines because we wanted to have cell lines available for people who wanted to do functional studies in patients, for example, in their fibroblasts, and also to be able to take them through the iPSC track to develop patient-specific cell lines of neurons and Schwann cells that could be used to test candidate treatments. So I think the main answer to Will's question is determine what you think the need is going to be for the biobank and how it's going to be used. And for myself, if I can't answer that question, then it limits my enthusiasm for it. Sandeep said that I mentioned the NIH Promise questionnaires. And he said they use the fatigue one, and these seem to be extensively studied. Is this why they are so preferable? So I want to answer you honestly that I don't consider myself an expert in the NIH Promise questionnaires. I think Sindhu Ramchandran from Michigan is really our expert there. But for me, I think of it as disease-specific questionnaires that are based on the NIH Promise approach. And so that these start out with large numbers of questionnaires. They get there's interactions with people who have CMT, and they get narrowed down, and the questionnaires get focused based on what the people with CMT think is important to them. And it's for that reason that they're so preferable to me. And Tobias asks, do you prefer symptom measures over quality of life? Given that patients' quality of life does not necessarily correlate with their symptoms. And again, the symptom measures that I'm talking about are parts of composite outcome measures, which I think have importance, particularly since for CMT, I think they're related to the biology of the disease. In other words, how much sensory loss is there distally, how much weakness is there distally. But I think that they don't necessarily correlate with the quality of life. And ultimately, what we're trying to do is develop treatments that improve people's lives with these diseases. So for reasons that I tried to explain before, I think that you need both. And if you do just one, then I think you're losing out. So I think Will is typing, so I'll wait for him to type. So Will is asking what was most eye-opening about meeting with industry. So I'm going to answer that in several ways, but I ask you to understand that part of my answers are based on the fact of my being very naive about this. So the first thing is to say that they were very interested in partnering and trying to develop treatments for these diseases. They cared about the diseases, and also CMT is really looking at molecular bases for demyelination and axonal loss. So I think that it's important for many industries to really, that makes it attractive because there are acquired demyelinating neuropathies, acquired demyelinating diseases, acquired axonal disorders that aren't CMT, but which lessons from this can actually be helpful for them. So I think that was one important eye-opening experience for me. The second eye-opening experience is just based on my bias going in, which was totally wrong. That I assumed that, oh, I'm an academic and I have a noble goal where industry is just in there to make money. That is totally not true. They were really professional, they're dedicated scientists, and the other thing is that they're really committed to developing these assays in a sophisticated, rapid fashion because they have to be able to screen just many candidate medications for many diseases. So things that took in a lab would take 2 years to look at like 100 compounds or something. They can do, they're already set up to do this in a very efficient way and they've had extensive troubleshooting to get rid of a lot of the potential errors in the system because their whole companies depend upon getting correct answers out efficiently and quickly. So just the quality of the people in industry and their ability to do things well has been eye-opening to me. And then they are just totally interested in seeing the patients and interacting with patients who have diseases. They're interested in doing the right thing for people. So those have all been eye-opening for me, and in large part just because of my ignorance and naivete. So what are possible strategies to deal with intellectual property issues? This is from Mario. When undertaking common research projects with industry. And I think that the answer here is to have experienced people from both sides and dealing with these issues up front. Because in the end, people, I think, have been willing to be fair. I'm not an expert in negotiating with industry, and the CMTA has been fortunate to have good people with experience in that. And I don't think anybody's been out to screw each other. I think it's been people just wanting to make sure things are clear up front. And so I think that having people with the appropriate expertise involved from the beginning with these is important. So Will says, it also seems like one thing that I've learned is it takes a long time. And I would answer that by saying, in order for this to actually work and bring treatment to patients, it has to be done correctly. It's taken me a long time because, in part, because I didn't have access to a course like this early on. And so I think it could have been done more quickly. I think that sort of related to that, for diseases like I'm involved with, it's really important to have large patient populations and to be able to work together. Because for the natural history of CMT1A, if I had tried to do this just out of my lab or my group, it never would have worked. But by having people from all around the world work together, train together, and analyze patients the same way, that's made it possible. So And sometimes it takes time to put that kind of a group together, but it's necessary. And this is a two-part question. Have you ever had a more modest request? Donate a drug you're looking for. I'm sorry. So I haven't had the type of request that donating a drug that I'm looking for a new population or disease to treat. Those things happen, but that's not, all I can do is talk about my experience. I think, so Andrea asked, does a company have any motivation for providing in-kind funding or other help for using a drug that is off patent? And I think the answer to that is yes, as long as the situation is de-risked. So that if you have the patient groups, if you can convince them that it can be looked at efficiently and that these components are in place, and I think they do have motivation. And providing in-kind funding is a little bit complicated because if you have an assay that they don't have, is perhaps they're willing to give you the medications, at least in a blinded way, to look at. But the way it's worked, for example, with Sanofi Genzyme, is that they have their in-kind funding has basically been to do the assays in-house, which are a major expense for them. And so it's in that sense that it's been in-kind. So Will is asking about the Orphan Drug Act and in the rare disease space. And that's something I ought to be able to answer, but I really can't because I just don't have any experience working with it. So I'm sorry, Will, I just can't give you an answer that's just not BS.
I think we're close to. Thank you for everyone joining us. Also wanted to say if you are in the evaluation form, if you box, it should take you to also copy the link to the browser. So our next webinar will be May 21st. Browse that training. Thank you, everyone, and I look forward to seeing you again on.
And thank you all for being part of a great course. Thanks.